Bismillahirrahmanirrahim. In the name of Allah, the most beneficent and merciful. Dear student, Assalamu alaikum. My name is Dr. Rana Mehmood, and today we are going to discuss about our third 3.4 lecture of Single Crystal X-Ray Diffraction, which is related to the course being studied in MS Chemistry Semester 2nd under the course code of Chem 5134. X-ray diffraction techniques. We were discussing about the softwares that are used in determining the structure of a single crystal in the previous lectures. Here are some other software that are also used which are related with the single crystal X-ray diffraction machine. This is known as SMART, Siemens Molecular Analytical Research Tools. The SMART Siemens Molecular Analytical Research Tool software control the operation of the diffractometer. It include equipment calibration, also include orientation matrix calculation, and the recording of the frames of different intensity data. Frames are typically measured in 3 or 4 batches. For omega scan data, within each batch, the psi and pi angles are fixed and the crystal is rotated around omega, normally in step of 0.3 degree. Different batches are collected with a different starting value of phi. This results in a measurement of series of closely spaced cross-section of reciprocal space. Hence, three-dimensional diffraction information is obtained. So this is the tool that is known as SMART, Siemens Molecular Analytical Research Tool, which is used to measure the diffraction. It also used to measure the calibration. It is also used in the orientation of the matrix calculation, and it also record the frame of diffracted intensity data. So this tool is very important in determining the structure of a single crystal, in which two angles are studied by using this tool. One is phi and other is chi. These two angles are also studied to determine the structure, and this result in measurement of series of closely spaced cross section of reciprocal space. In total, up to 1200 frames are normally acquired, which corresponds to a 2 theta coverage of 0 to 55 degree. But these limits may be changed if necessary. For example, if more data is required, then typical program frame for a strongly diffracting crystal is shown below. The vertical color bar is used to adjust the contrast to optimize the observed diffraction pattern. And a movie showing the diffraction pattern at the crystal is rotated in a sequence of 44 frames that is available as a MPEG file 403 kilobyte or in Macintosh QuickTime format, that is 735 kilobyte. For different operating system used in the computer, there are different kind of software and different kind of frame values that are used in MPEG files and also in Macintosh quick format. Here is a pictorial display of the frame that is comes out when we use the smart tool, and all about this is discussed in the previous slide. Saint Simon area detector integration. This is an other software tool that is used in single crystal X-ray diffraction technique. The Saint, that is Siemens area detector integration program. It is used to integrate the raw frames data from the area detector and produce a scaled and merged set of reflection intensities. The corrections for the absorption effects and crystal decay are possible, and as well as the merging of data from different collection batches or different crystal is also studied using this Saint Siemens area detector integration software. The integration can be performed concurrently as the frames are being collected, so that reflection data file is available for structure solution immediately after the frame data collection has finished. So this is another software programming which is used in the single crystal XRD, and this programming is used by using computer which are attached with the machines after operating machine mean XRD instrument, which provides the different kind of scattering pattern, diffraction patterns, and by using those diffraction pattern, these different kind of softwares are used to obtain the real crystal image from that single crystal. Solving a crystal structure by single crystal diffraction technique. So, solving single crystal structure by the diffraction technique, the crystal must be a single crystal. And the Bragg equation specifies that if a crystal is rotated within a monochromatic X-ray beam such that every conceivable orientation of the crystal relative to the beam is achieved, each set of planes will have had the opportunity to satisfy the Bragg equation and will have given rise to reflection. In order to solve a crystal structure, it is necessary to record a large number of reflections. This implies accurately measuring their intensities and recording their direction with respect to crystal orientation and initial X-ray beam direction. Many experimental techniques have been devised to achieve this. The steps involved in the crystal structure determination are summarized in the flow chart. So in this slide, we studied that if we want to solve a crystal structure by a single crystal diffraction technique, the crystal must be a single crystal. And this is solved by using the Bragg equation, which specifies that a monochromatic X-ray beam of conceivable orientation of crystal is achieved, and the set of planes is also specified by using the Bragg equation. And to solve the crystal structure, it is also necessary to record large number of reflections that accurately measures their intensities and also their direction with respect to crystal orientation. Many experimental techniques have been devised to achieve this. This is a schematic diagram in which we select a suitable crystal first, and then the crystal system and the unit cell dimensions are studied. Then full data set collection is measured, and after that previous lattices are observed, and then space group is found. Construction of an electron density map is required. After that, the location of the atom or position is studied, and at the end, structure refinement comes. When we move on towards the application of single crystal X-ray diffraction, this kind of diffraction is most commonly used for precise determination of a unit cell. It includes cell dimension and position of atom within the lattice. Bond length and bond angles are directly related to the atomic positions. So, the crystal structure of a mineral is a characteristic property that is the basic for understanding many of the properties of each mineral. Specific applications of single crystal diffraction include the following. Number 1, new mineral identification and crystal solution and refinement. This is one of the application of single crystal X-ray diffraction. That is, we can identify the new mineral and we can solve the crystal, and also we can refine its structure. Second is the determination of the unit cell, bond length, bond angles, and site ordering. By using this single crystal X-ray diffraction technique, we can determine the unit cell of that crystal we can also determine the bond length, the distance between the atoms, and the bond angles at which the certain atoms are attached to one another and their site ordering. And the characterization of the cation and anion coordination is also one of the application of single crystal X-ray diffraction. And the variation in crystal lattice with chemistry is also studied by using SCXRD, Single Crystal X-ray Diffraction technique. Dear student, there are some other applications of single crystal X-ray diffraction technique. By using this technique, if we have some specialized chambers, the structure of high pressure and or low temperature phases can be determined by using this technique. And also the determination of the crystal chemical versus environmental control on the mineral chemistry can also be studied by using this technique. And the powder patterns can also be derived from single crystals by the use of specialized cameras. These cameras are known as GAN or fill. So here we are going to discuss the strength and limitation of single crystal X-ray diffraction. The strength and the limitation of the single crystal X-ray diffraction are as follows. First, we are going to discuss the strength of this technique, and then after that we will have a look on some limitations that occurs using this technique. The strength of this technique is no separate standard are required, and we know that this technique is a non-destructive technique. That means the sample that we used in this technique, that sample is not destroyed. We can use that sample again and again because this technique is a non-destructive technique for the sample. And when we move on towards the limitation, it has that it must have a single robust stable sample, generally between 50 to 250 microns in size. This is the limitation, one of the limitation, that if we don't have single crystal, then this technique will not work to determine the structure of that crystal lattice. And for this we would have to be in the range of size from 50 to 250 microns. Optically clear sample is required, and the twinned samples can be handled with difficulty. So if the samples are twin, then these could be handled, but it will be very difficult to handle them. And the data collection generally require between 24 and 72 hours. So it takes time to collect the data and then to solve the data to determine the structure of a single crystal, and the time required for that data collection and the solution of that data is 24 to 72 hours. And when we discuss the advantages and disadvantages of X-rays, the X-rays that are used in the single crystal X-ray diffraction technique, these X-rays strike the single crystal, and after striking, these are scattered at different angles and make a diffraction pattern. That diffraction pattern helps us to determine or to study the structure of that crystal. So first advantage of using the X-rays, that X-ray is the cheapest, the most convenient and widely used method. That's why this X-ray is used in single crystal X-ray diffraction technique. And the second advantages of using this X-ray technique is X-rays are not absorbed very much by air. So the specimen need not to be in a evacuated chambers. So if the sample that is being observed is present in an open air environment, then these X-rays can easily be used to observe the specimen, because X-rays are not absorbed by the air surrounding the sample. And the advantages are these, and then when we discuss the disadvantages of the X-rays, these are that they do not interact very strongly with lighter elements. So this is one of the main disadvantage of the X-ray analysis, that when we are studying the lighter elements, then it makes us difficult to study because they don't interact very strongly with that lighter elements. The main limitation of the method is the requirement of a pure crystal of high regularity. So this is the other disadvantage, that the method is required of a pure crystal of high regularity if we use the X-rays as a source to determine the single crystal structure. Here is a schematic diagram that is also showing the comparison between the single crystal and the powder diffraction. In the single crystals, source used is X-ray source, and that X-rays are fallen on a single crystal, and after that these X-rays are scattered at different angles and make a diffraction pattern. After studying this diffraction pattern, there is another uh, electron density map comes, and that electron density maps help us to determine the structural model of that single crystal. So the structure, solution and refinement occurs after the diffraction pattern obtained by the machine, and this diffraction pattern is taken to the computer, and different kind of software that we have studied are used to determine the electron density maps and then to determine the structural mode. And the other method that is used for the structure determination, that is powder diffraction. In the powder diffraction, same source X-rays are used and a single crystal is taken, and we obtain a graph which is showing some peaks, and this graph is then, by using some different kind of software, is converted into determine the structure of the whole crystal system. Here is some comparison between X-ray diffraction Px Rd versus single crystal X-ray diffraction SCXRD. Dear student, here we are going to compare PXRD and SCXRD, powder X-ray diffraction technique and single crystal X-ray diffraction technique. As we know that powder X-ray diffraction is not as powerful as single crystal X-ray diffraction technique. And powder X-ray diffraction gives less accurate atomic position as compared to SCXRD. Single crystal X-ray diffraction requires the growth of a single crystal while there is no need for PXRD. So this is the advantage of PXRD, that there is no need to have a single crystal. It can be used by using the whole crystal. In powder X-ray diffraction, sample must be in powder form, while in single crystal XRD, only single crystal is required. In powder X-ray diffraction technique, large crystalline size and non-random crystals orientation both lead to peak intensity variation. And powder X-ray diffraction obtains diffraction pattern for bulk material of the crystalline solid, while single crystal XRD does not necessarily represent the overall material. In powder XRD diffraction pattern is obtained from a powder of material rather than in single crystal XRD. Individual crystals give the diffraction pattern. Powder XRD is easier and more convenient than single crystal XRD, as it does not require individual crystal to be made. An X-ray single crystal will be scattered only in certain directions when they hit the formed lattice plane found by the atoms. This will cause high intensity peaks. For polycrystalline material, X-ray will be scattered in many directions, leading to a large bump distributed in a wide range to theta instead of high intensity narrow peaks. So this is all about the comparison between powder XRD and single crystal XRD. Thank you very much, dear student. Now we will come with the next lecture about this single crystal. Till then, Allah Hafiz.